So, here's something to think about as you settle in tonight. Right now, in this exact moment, you are not alone. And I don't mean that in some poetic, philosophical way. I mean it literally. Right now, as you lie there are roughly 39 trillion microorganisms living on you and inside you. Bacteria, fungi, archaea, viruses. 39 trillion. That number is so large it almost stops meaning anything. So let me put it another way. The number of microbial cells in your body is roughly equal to the number of human cells. Some estimates put it slightly higher. For every cell in your body that carries your DNA, there is approximately one cell that carries somebody else's entirely. You are, in a very real sense, a walking ecosystem, a coral reef in pajamas. And the vast majority of these organisms are not just harmless, they're essential. They're digesting your food, training your immune system, producing vitamins, protecting you from pathogens, influencing your mood through chemical signals sent to your brain. You couldn't survive without them. You and your microbes are partners in an arrangement that goes back hundreds of millions of years, long before anything that looked even remotely human walked the earth. And yet, for most of human history, nobody had the faintest idea any of this was happening. The entire microbial world, this vast, teeming, astonishingly diverse kingdom of life, was completely invisible, hidden in plain sight, in every drop of water, every handful of soil, every breath of air, every surface you've ever touched. And the story of how we discovered it, how we slowly learned to see it and understand it, is one of the most fascinating stories in all of science. That's what we're going to talk about tonight. Microbiology. The whole sprawling, beautiful, sometimes terrifying history of it. We're going to start at the very beginning, before anyone even knew microbes existed, and work our way forward through centuries of discovery, rivalry, breakthroughs, catastrophic mistakes, and moments of sheer brilliance. We'll take our time. There's no rush at all. If you drift off somewhere along the way, that's perfectly fine. The microbes will still be there in the morning. They've been around for about 3.5 billion years. They can wait. Let's start with the world before anyone could see them. For thousands of years, people got sick, and nobody really understood why. They had theories, of course. The ancient Greeks and Romans believed in something called miasma, which was essentially the idea that disease came from bad air, foul-smelling vapors rising from swamps, rotting organic matter, sewage. If the air smelled bad, it could make you sick. And you know what? In a practical sense, this wasn't completely useless. If you avoid areas that smell like sewage and rotting things, you will, in fact, avoid some diseases. Not because of the smell itself, but because those environments happen to harbor the actual causes of disease. It was the right conclusion reached, for entirely the wrong reason, which happens more often in the history of science than you might think. But miasma theory had real consequences. It shaped public health for centuries. When the Black Death swept through Europe in the 14th century, killing somewhere between 75 and 200 million people, the prevailing medical advice was to avoid bad smells. Doctors wore those eerie beaked masks stuffed with flowers and herbs, trying to filter the poisonous air. Cities burned aromatic woods. People carried pomanders, little balls of perfume, hoping to ward off the plague. None of it worked, obviously, because the plague wasn't carried by bad air. It was carried by Yersinia pestis, a bacterium transmitted by fleas on rats. But nobody would understand that for another 500 years. And that's the thing about the microbial world. It was always there, always shaping human history in profound ways, causing pandemics, spoiling food, fermenting wine, curdling milk, rotting wood. People could see the effects everywhere. They just couldn't see the cause. It's like living your entire life feeling the wind, but having no concept of air. You know something is happening, but you can't point to the thing doing it. And this went on for millennia. Until one day, in the city of Delft, in the Dutch Republic, in the late 1600s, a man with no scientific training, no university education, no connections to the academic world, looked through a tiny glass lens he had ground himself, and he saw them. He saw the invisible world, and nothing would ever be the same. His name was Antony van Leeuwenhoek, born in 1632, in Delft, the same city where Johannes Vermeer was painting his luminous canvases around the same time. Think about that for a second. In the same small Dutch city, at the same moment in history, one man was revealing the hidden beauty of light and color on canvas, and another was revealing a hidden world of living creatures too small for the human eye to see. Delft must have been something else in the 17th century. Leeuwenhoek was a draper, a cloth merchant. He sold fabrics. That was his profession. He wasn't a scientist. He wasn't a doctor. He hadn't studied natural philosophy at any university. But he had a skill that turned out to be extraordinary. He was phenomenally good at grinding lenses. Now, in the textile trade, magnifying lenses were used to inspect the quality of cloth, to count the threads per inch, to check for imperfections in the weave. It was a practical tool of the trade. But Leeuwenhoek took it much, much further. He became obsessed with making lenses, and through years of practice and experimentation, he developed techniques for grinding tiny, nearly spherical lenses of astonishing quality. His best lenses could magnify objects by about 270 times. Some historians think he had secret methods he never shared with anyone. He certainly never published his lens grinding techniques, and when he died in 1723, at the age of 90, his best lenses effectively died with him. Nobody else could make them as well for decades afterward. And here's what's important to understand. When we say microscope, you're probably picturing something like the compound microscope you might have used in a biology class, with its tube and its eyepiece and its objective lenses stacked in a turret. Compound microscopes existed in Leeuwenhoek's time. Robert Hooke, over in England, used one to study cork and coined the word cell in 1665 when he saw the tiny box-like structures in the cork tissue. But the compound microscopes of the 17th century had a serious problem. They used multiple lenses, and each lens introduced optical distortions, chromatic aberration, blurriness. The images were fuzzy, unreliable. Leeuwenhoek's microscopes were different. They were incredibly simple. Just a single tiny lens, mounted in a small brass or silver plate, with a pin to hold the specimen, and a set of screws to adjust the position and focus. You held the whole thing right up to your eye, almost touching it, and peered through the lens like looking through a keyhole into another world. They were awkward to use, uncomfortable even, but because there was only one lens, and because that lens was ground with such extraordinary precision, the images were sharp. Remarkably, astonishingly sharp. And what Leeuwenhoek saw through those lenses changed everything. In 1674, he looked at a drop of lake water from a nearby body of water called the Berkelse Meer, and he saw tiny living creatures swimming around. He called them animalcules, little animals. Some of them were probably what we now call protozoa, single-celled organisms like Paramecia and Vorticella. He described their movements, their shapes, their behaviors with a detail and accuracy that still impresses biologists today. He drew them, he measured them, estimating their sizes by comparing them to grains of sand. He watched them eat, divide, and die. He was seeing, for the first time in human history, organisms that no human eye had ever perceived. But he didn't stop there. Over the next several decades, Leeuwenhoek examined everything he could get his hands on. He looked at rainwater, well water, seawater, infusions of pepper and ginger, water from gutters and ditches. He looked at blood and saw the red blood cells, which he described in remarkable detail. He looked at sperm cells and described them so precisely that the Royal Society in London blushed a little when his letters arrived. He examined the legs of lice, the structure of hair, the scales of fish skin. He was insatiable. And then, in 1683, he did something that might be his most consequential observation of all. He scraped the white stuff from between his own teeth, mixed it with a little clean rainwater, and looked at it under his lens. And there they were, bacteria. He saw bacteria. He described them as tiny rods, spirals and spheres, moving through the water with a liveliness that astonished him. He described some of them shooting across his field of view, like a pike darting through water. Others tumbled and spun. He noted that there were more of these tiny organisms in his own mouth than there were people in the whole of the Netherlands. These were among the first observations of bacteria in history, and he was finding them in his own mouth. Now, Leeuwenhoek didn't have anyone nearby to share these discoveries with in any scientific capacity. He wasn't part of a university. He didn't attend scientific meetings. But he had a contact, a physician and anatomist named Regnier de Graaf, who knew about Leeuwenhoek's exceptional lenses and wrote to the Royal Society of London, the most prestigious scientific body in Europe, recommending that they pay attention to this Dutch draper. And so Leeuwenhoek began writing letters to the Royal Society. Over the course of his long life, he wrote more than 550 letters, written in Dutch because he didn't know Latin or English, describing in meticulous detail everything he saw through his lenses. The letters are extraordinary documents, combining precise observation with genuine wonder. He wasn't just recording data, he was marveling at it. And the Royal Society was initially skeptical, as you might expect. This wasn't how science was supposed to work. The man had no credentials, no formal training. Some of his claims seemed outlandish, but they sent a delegation to Delft to verify his observations, and when they looked through his microscopes, they saw exactly what he had described. Every last detail checked out. He was elected a Fellow of the Royal Society in 1680, one of the most unlikely members in its history. A cloth merchant from Delft, sitting alongside Newton and Hooke and Boyle. Here's what I find so wonderful about Leeuwenhoek. He had no theory. He wasn't trying to prove anything. He wasn't motivated by a hypothesis about disease or biology. He was just relentlessly, insatiably curious. He wanted to look at things, everything, anything. And because he looked with such care and such honesty, he saw what was actually there. No preconceptions, no agenda, just an eye, a lens, and an open mind. And what he found was a world that nobody had imagined. A world teeming with life at a scale too small to see. A world that had been right there, all along, in every drop of water, in every human mouth, in every patch of soil. But here's the thing. Leeuwenhoek discovered these tiny organisms, and he described them beautifully, and the scientific world acknowledged their existence. And then, for quite a long time, nobody really knew what to make of them. What were they? Where did they come from? Were they alive in the way that animals and plants were alive? Did they just appear spontaneously, whenever conditions were right, springing into existence from non-living matter like magic? Or did they come from other organisms like themselves, reproducing and multiplying in some miniature echo of the way larger creatures reproduce? This question, the question of where microbes come from, would ignite one of the longest, most contentious, and most important debates in the history of biology. It would take nearly 200 years to settle, and the story of how it was settled involves some of the most elegant experiments ever designed, a stubborn genius with a perfectionist streak, and a controversy that reached all the way to the French emperor. But the roots of the argument go back much further than Leeuwenhoek, all the way to ancient Greece, to an idea that had seemed perfectly obvious to brilliant minds for over 2,000 years, and that idea was spontaneous generation, the belief that living things could arise from non-living matter. And before you dismiss this as obviously ridiculous, consider how reasonable it must have seemed for most of human history. You leave meat out in the open, and within days, it's crawling with maggots. Where did they come from? You didn't put them there. You didn't see anything arrive. They just appeared, seemingly born from the rotting flesh itself. Leave grain in a dark barn, and mice show up. Leave old rags in a corner with some wheat, and the alchemist Jan Baptist van Helmont claimed in the 17th century that you'd get mice. Just wait 21 days, he said, and the mixture of rags and wheat would generate mice. He wrote this down as a recipe for making mice from rags. It sounds absurd to us, but Van Helmont was a serious scholar, and he was simply recording what everyone believed they observed. Mud produces frogs. Standing water produces mosquitoes. Decaying matter produces flies. Life, it seemed, could spring from the inanimate world whenever conditions were right. Aristotle believed in spontaneous generation. He wrote about it quite clearly. He thought certain animals arose from putrefying matter, that eels came from mud, that flies came from rotting fruit. And Aristotle was not a fool. He was one of the most careful observers in the ancient world, a man who dissected dozens of species and catalogued their anatomy with extraordinary precision. But he was working without a key piece of information, the same piece that everyone lacked until Leeuwenhoek. He couldn't see the tiny organisms and eggs that were actually responsible, so the idea persisted. For century after century, it seemed self-evidently true. Life comes from death. New creatures spring from old matter. It was woven into the intellectual fabric of Western thought so deeply that questioning it felt almost perverse. The first serious challenge came from an Italian physician named Francesco Redi in 1668. Redi was a fascinating character, a poet, a linguist who spoke at least 6 languages, a naturalist, and the personal physician to the Grand Duke of Tuscany. He was, by all accounts, both charming and fearless in intellectual debate. And he was skeptical about spontaneous generation, at least for larger organisms like flies. So he designed an experiment that was elegant in its simplicity. He took several jars and placed pieces of meat in each one. Some jars he left open, with nothing covering them at all. Some he sealed tightly with wax. And some he covered with fine gauze, a mesh material, fine enough that air could pass through freely, but insects could not. Then he waited, and he watched. The results were clear and devastating. The open jars, the ones flies could freely access, developed maggots, lots of them. The sealed jars, no maggots, not a single one. And the gauze-covered jars, this is the clever part, no maggots either, even though the meat had full access to air, even though it rotted just as thoroughly as the meat in the open jars. But Redi could see flies landing on the gauze, trying to get to the meat, and occasionally leaving their eggs on the outside surface of the gauze. Maggots would sometimes appear on the gauze but never on the meat beneath it. Redi had demonstrated that maggots didn't come from the meat. They came from fly eggs. No flies, no eggs, no maggots. The meat could rot all it wanted, and it would never spontaneously produce maggots on its own. This was a beautiful experiment. Clean, controlled, convincing. One of the first truly well-designed controlled experiments in the history of biology, actually. And it did change a lot of minds about larger organisms. People largely accepted that maggots and mice and frogs didn't arise spontaneously. But here's where it gets complicated. When Leeuwenhoek's animalcules entered the picture, the whole debate reignited with fresh fury, because these were different. These were so unimaginably small, so seemingly simple, that many people argued they must arise spontaneously. How could something that tiny reproduce? How could something you can barely see have parents? Maybe the rules were different at that scale. Maybe the very smallest forms of life really did spring from broth and water and decaying matter. And so began a debate that would rage for the next century and a half. In one corner, you had people like John Needham, an English naturalist and Catholic priest, who in 1745 performed experiments that seemed to support spontaneous generation. He boiled mutton broth to kill anything living in it, poured it into flasks, sealed them with corks, and waited. After a few days, the broth was cloudy with microorganisms. "See," he said, "they must have arisen spontaneously, because I killed everything by boiling and then sealed the flask." In the other corner was Lazzaro Spallanzani, an Italian priest and scientist, a man of enormous energy who was also one of the finest experimental biologists of the 18th century. In 1768, Spallanzani challenged Needham's results directly. He argued that Needham hadn't boiled his broth long enough, and that his cork seals weren't airtight, allowing contamination from outside. So Spallanzani boiled his broths for much longer, up to an hour, and sealed his flasks by melting the glass necks shut while they were still hot, fusing them into airtight containers. And his broths stayed clear. No growth. Nothing. Week after week. Nothing. This strongly suggested that the microbes in Needham's experiments had come from the outside, from contamination, not from the broth itself. But Needham had a clever rebuttal, and this is where the debate gets really interesting. He said that by sealing the flasks so tightly and boiling for so long, Spallanzani had destroyed something essential in the air, some vital force, some life-giving property that was necessary for spontaneous generation to occur. Without this vital force, of course, nothing would grow. It wasn't that spontaneous generation didn't happen. It was that Spallanzani had prevented it by removing the very thing that made it possible. And honestly, this was a reasonable objection given what they knew at the time. It was wrong, but it was genuinely reasonable. Nobody understood what air was made of, or what role, if any, oxygen played in life. The concept of a vital force in air wasn't pseudoscience in the 1800s. It was a legitimate hypothesis, and so the debate was stuck, deadlocked. Each side could explain away the other's results. What was needed was an experiment so clever, so airtight in its logic, so beautifully designed that nobody could wriggle out of the conclusion. And that experiment wouldn't come for almost another century, until a Frenchman with a famously combative personality and an obsessive attention to detail entered the arena. Louis Pasteur. Born in 1822 in Dole, in the Jura region of eastern France, the son of a tanner who had served as a sergeant major in Napoleon's army. Pasteur was not a biologist by training. He was a chemist. His early work was on crystal structures, specifically on the optical properties of tartaric acid, and he made a genuinely important discovery about molecular asymmetry, about how certain molecules exist in mirror image forms. This was technically brilliant work, and it might seem, on the surface, to have nothing to do with microbes. But Pasteur had a quality that would define his entire career and make him one of the most consequential scientists who ever lived. He was relentlessly practical. He didn't do science in a vacuum. He was constantly drawn to real-world problems, industrial problems, problems that mattered to ordinary people, and this tendency would pull him, step by step, from chemistry into biology, from crystals into the living world, and eventually into the most consequential scientific battles of the 19th century. The path that led Pasteur to microbiology began, of all places, with alcohol. In the 1850s, Pasteur was a professor at the University of Lille in northern France, a city with a thriving beet sugar industry that also produced alcohol through fermentation. And the local manufacturers had a problem. Sometimes their fermentations went wrong. Instead of producing nice, clean alcohol, the beet juice would turn sour, producing lactic acid instead of ethanol. Batch after batch was ruined, and the financial losses were considerable. And they asked Pasteur, the local chemistry professor, if he could figure out what was going wrong. Now, at the time, the prevailing view among chemists, championed most forcefully by the great German chemist Justus von Liebig, was that fermentation was a purely chemical process. Yeast might be present during fermentation, but it wasn't doing anything important. The yeast were a byproduct, not a cause. Fermentation was just a chemical decomposition, like rust forming on iron. Liebig was one of the most influential chemists of the 19th century, and his authority on this point was considerable. Pasteur looked at the problem differently. He examined the healthy fermentation vats under the microscope and saw plump, round, budding yeast cells. Then he examined the sour vats, the ones producing lactic acid, and found something different. Tiny, rod-shaped organisms that were not yeast at all. They were bacteria, though that word wasn't commonly used yet. And Pasteur made the bold claim that fermentation was not a purely chemical process. It was a biological one. Living organisms, specific living organisms, were responsible for specific chemical transformations. Yeast produced alcohol. These other bacteria produced lactic acid, different microbes, different products. Fermentation was life at work. This was a radical claim, and Liebig pushed back hard. The debate between Pasteur and Liebig over the nature of fermentation lasted years and got quite heated. But Pasteur was right, and his evidence was meticulous. He showed that you could kill the microbes by heating the liquid, and fermentation would stop. He showed that you could introduce specific organisms into sterile broth and get specific, predictable chemical products. And out of this work came a practical innovation that still bears his name: pasteurization, the process of heating a liquid to a specific temperature for a specific time to kill harmful microbes without significantly changing the flavor. It was developed to save the French wine and beer industries, and today it protects billions of people from foodborne illness every day. Every carton of milk, every bottle of juice you've ever drunk was almost certainly pasteurized, using a process invented by a chemist who was trying to help French brewers make better beer. But here's where it all connects. If specific microbes cause specific chemical processes like fermentation, then the question of where microbes come from wasn't just an abstract philosophical puzzle anymore. It was a practical question with enormous implications. If microbes arose spontaneously from non-living matter, then you could never truly prevent contamination. New microbes would just keep appearing out of thin air. But if microbes only came from other microbes, if they reproduced like everything else, then you could control them. You could keep them out. You could sterilize things and they would stay sterile. The answer mattered. In 1859, Pasteur designed his most famous experiment, and it was, in its way, a masterpiece of scientific thinking because it addressed the one objection that had protected spontaneous generation for over a century. Remember Needham's argument against Spallanzani, that by sealing the flasks, you cut off the vital force in the air? Pasteur's solution was the swan neck flask. He took ordinary glass flasks, filled them with a nutrient broth, and then heated and bent the necks into long, curving S-shapes, like the neck of a swan. The crucial detail was that the necks were left open at the tip. Air could flow freely in and out. Whatever vital force the air contained was not excluded, but the curved neck acted as a trap. Dust particles and microorganisms carried in the air would settle in the low bends of the neck, caught by gravity, and never reach the broth sitting in the body of the flask. He boiled the broth to sterilize it, let it cool, and waited. Days passed, weeks, months. The broth remained perfectly clear, crystal clear, no microbial growth whatsoever. Air was getting in, flowing freely through the open neck. The supposed vital force was present, but nothing grew. And then, the decisive move, the coup de grace. Pasteur took some of the flasks and tipped them, allowing the broth to flow up into the curved neck and back down, picking up the trapped dust and particles that had settled in the bends. Within days, those flasks were cloudy with microbial life. The organisms hadn't come from the broth, they hadn't arisen spontaneously. They had come from the air, carried on dust particles, and the swan neck had kept them out. It was devastating. It was elegant. And it was essentially the end of spontaneous generation as a serious scientific idea. Pasteur presented his results to the French Academy of Sciences in 1864, in a public lecture at the Sorbonne. That was, by all accounts, a triumph. The audience included not just scientists but politicians, literary figures, members of high society. Pasteur was a gifted showman. He knew how to build tension, how to present results for maximum dramatic impact, how to make science feel like theater, and he reportedly declared, with characteristic confidence, that the doctrine of spontaneous generation would never recover from this mortal blow. He was right. Some of those original swan neck flasks, by the way, are still preserved at the Pasteur Institute in Paris, and the broth in them is still clear, more than 160 years later. But Pasteur wasn't done, not even close. Because disproving spontaneous generation was just one step on a much larger journey. If microbes only come from other microbes, and if specific microbes cause specific chemical processes, then maybe, just maybe, specific microbes also cause specific diseases. This idea, the germ theory of disease, had been floating around in various forms for centuries. The Italian physician Girolamo Fracastoro had proposed something remarkably similar back in 1546, suggesting that disease could be transmitted by tiny, invisible seeds he called seminaria. But nobody had been able to prove it. Nobody had the tools or the techniques to connect a specific organism to a specific disease with rigorous evidence. Until Pasteur, and until a quiet, methodical German country doctor named Robert Koch, who would become Pasteur's greatest rival and, in some ways, his most important complement. Because where Pasteur was flamboyant, Koch was precise. Where Pasteur relied on intuition and bold leaps, Koch was precise. Koch developed rigorous, systematic methods that would become the gold standard for identifying disease-causing organisms for a century to come. And the tension between these two men, the Frenchman and the German, working in parallel, sometimes grudgingly acknowledging each other's achievements, more often attacking each other through publications and proxies, fueled by scientific ambition and by the still raw wounds of the Franco-Prussian War of 1870, that tension drove microbiology forward at a pace that might not have been possible otherwise. Sometimes rivalry produces better science than collaboration. This was one of those times. Robert Koch was born in 1843 in Clausthal, a mining town in the Harz Mountains of Germany. He was one of 13 children and reportedly taught himself to read using newspapers before he started school. His father was a mining official, a man of modest means and practical temperament. Koch grew up quiet, meticulous, and possessed of an almost frightening patience. He could sit at a microscope for hours upon hours, days upon days, observing, recording, sketching, waiting for something to reveal itself. He was the anti-Pasteur in temperament, in personality, in almost every way except brilliance. While Pasteur came to microbiology through chemistry, Koch came to it through medicine. After studying medicine at the University of Göttingen, he became a country doctor, a district medical officer in a small town called Wolstein, in what is now Poland. He treated farmers and their families, dealt with the everyday ailments of rural life, set bones, delivered babies. But in his spare time, using a microscope that his wife Emmy had bought him as a birthday present, he began studying anthrax, and this is where his story really takes off. Anthrax was a devastating disease of livestock, and it was common in rural Germany and France. Cattle and sheep would fall sick and die suddenly, sometimes within hours, their blood turning dark and thick. It was economically ruinous for farming communities, and occasionally, it killed people too, especially those who handled infected hides or wool. And Koch, working essentially alone in a makeshift laboratory set up in his own home, with his birthday present microscope and whatever equipment he could cobble together, did something remarkable. He isolated the anthrax bacillus, a large rod-shaped bacterium, from the blood of infected animals. He grew it in pure culture, outside of any animal body, using the aqueous humor from ox eyes as a growth medium. He observed the bacteria forming spores, tough, dormant survival structures encased in protective coats that could persist in soil for years, even decades, explaining why anthrax kept recurring in certain fields long after infected animals had been removed. And then, critically, he took the cultured bacteria and injected them into healthy mice, which promptly developed anthrax and died. He recovered the same bacteria from the dead mice. Think about what Koch had done here. He had taken a specific organism out of a sick animal, grown it in pure isolation, with nothing else present, put it into a healthy animal, reproduced the disease perfectly, and recovered the same organism again. He had closed the causal loop. He had demonstrated, with a rigor that nobody could argue with, a direct link between a specific microbe and a specific disease. This was in 1876, and Koch was 33 years old, a country doctor nobody had heard of. When he presented his work to Ferdinand Kohn, a prominent botanist at the University of Breslau, Kohn immediately recognized its importance and helped Koch publish. And when Pasteur's collaborator Émile Roux brought the news back to Paris, even Pasteur was impressed, though he would never have said so publicly. Koch went on to formalize this approach into what we now call Koch's postulates, a set of 4 criteria that had to be met before you could claim that a particular microorganism caused a particular disease. First, the organism must be found in all cases of the disease, but absent from healthy individuals. Second, it must be isolated from a diseased individual and grown in pure culture. Third, when that pure culture is introduced into a healthy susceptible host, it must reproduce the disease. And fourth, the organism must be re-isolated from the experimentally infected host and shown to be identical to the original. These postulates are one of the most important contributions in the entire history of medicine. They gave scientists a rigorous framework, a checklist, a set of standards for proving causation. Before Koch, claims about disease were often vague, anecdotal, based on correlation rather than proof. After Koch, there was a method, a protocol, a standard of evidence, and it worked. It was demanding, but it worked. Koch's greatest triumph came in 1882, when he identified the bacterium that causes tuberculosis. Now, tuberculosis was not some minor rural disease. It was the leading cause of death in Europe. 1 in every 7 Europeans was dying of TB. They called it the White Plague, consumption, the captain of all these men of death. It was everywhere, in every city, in every class, and nobody knew what caused it. Many physicians thought it was hereditary, a weakness passed down through family lines. Others thought it was caused by bad air, damp conditions, poor nutrition, moral failings. The idea that it was caused by a specific, identifiable, transmissible organism was far from universally accepted. Koch changed that, and the way he did it was a triumph of technical innovation as much as scientific insight. The tuberculosis bacillus, Mycobacterium tuberculosis, is notoriously difficult to work with. It grows agonizingly slowly compared to most bacteria, taking weeks to form visible colonies where other bacteria would grow overnight. And it has a waxy outer coat that resists the standard staining techniques of the era, making it nearly invisible under the microscope. Koch solved both problems. He developed new staining methods, using alkaline dyes and a counterstain that made the bacilli visible as tiny blue rods against a brown background. He developed new solid culture media, using coagulated blood serum that allowed the organism to grow in the laboratory. And on March 24, 1882, he presented his findings to the Berlin Physiological Society in a lecture that is remembered as one of the most important in the history of medicine. He laid out the evidence with devastating thoroughness. He showed the bacillus in the tissues of TB patients, in the lungs, in the lymph nodes. He showed it growing in pure culture on his blood serum medium. He showed it causing tuberculosis in guinea pigs and rabbits that he had experimentally infected. He showed it re-isolated from those animals. Every one of his postulates satisfied. Link after link in the chain of evidence, each one unbreakable. The audience sat in stunned silence when he finished. There was no applause at first, just silence, as the implications sank in. Paul Ehrlich, who was in the audience that night and would later win a Nobel Prize himself for his work on immunity, later described it as the most significant experience of his entire scientific life. Koch had proved that the most feared disease in the world, a disease that killed millions, was caused by a tiny rod-shaped bacterium no bigger than a few thousandths of a millimeter. And in doing so, he had demonstrated that the germ theory of disease wasn't just an interesting hypothesis, not just a good idea. It was real. It was provable. It was the future of medicine. Now, I mentioned that Pasteur and Koch were rivals, and here's where that rivalry gets really interesting. While Koch was the methodical detective, identifying the criminal and building an airtight legal case, Pasteur was more like the engineer, looking for ways to fight back, to use the knowledge of germs to actually do something about disease. And Pasteur's great weapon was vaccination. The concept of vaccination wasn't new. Edward Jenner had developed the smallpox vaccine back in 1796, using cowpox, a mild disease of milkmaids, to protect against the deadly smallpox. But Jenner's method was specific to one disease, and nobody really understood why it worked. It was an empirical observation, not a generalizable principle. Pasteur figured out the underlying principle. Working with chicken cholera in the late 1870s, he made a serendipitous discovery that changed the trajectory of medicine. His laboratory assistant, Charles Chamberlain, had accidentally left a culture of the chicken cholera bacterium sitting on a bench over the summer holiday while the lab was closed. When they returned in the fall and injected chickens with this old, stale, weakened culture, the chickens got mildly ill but recovered completely. And then, the critical moment, when those same chickens were later injected with fresh, virulent, full-strength bacteria, they didn't get sick at all. They were immune. Pasteur grasped the significance immediately. The weakened, attenuated organisms had trained the chickens' immune systems to recognize and fight the pathogen. And this principle, this idea of using weakened or killed organisms to stimulate immunity, could be applied to any infectious disease, not just smallpox. He developed vaccines for anthrax, demonstrated famously in a dramatic public trial at Pouilly-le-Fort in 1881, where vaccinated sheep survived an anthrax challenge that killed all the unvaccinated controls. And then, most dramatically of all, he developed a vaccine for rabies. The rabies vaccine story is one of the most gripping in the history of medicine. Rabies was a terrifying disease, almost always fatal once symptoms appeared, and the death it caused was agonizing. Pasteur had been working on the disease for years, growing the virus in the brains of rabbits and weakening it through serial passage. In July 1885, a 9-year-old boy named Joseph Meister was brought to Pasteur's laboratory. The boy had been badly mauled by a rabid dog, bitten 14 times. Without treatment, he would almost certainly die an agonizing death. Pasteur was not a physician. He was not licensed to practice medicine. Administering an untested vaccine to a child was a massive personal and professional risk. But after consulting with physicians who confirmed that the boy's prognosis was hopeless without intervention, Pasteur began the course of injections. Over 11 days, the boy received 13 injections of progressively stronger rabies preparations. And Joseph Meister survived. He never developed rabies. He grew up, lived a long life, and eventually became the gatekeeper at the Pasteur Institute in Paris. The news spread around the world. People bitten by rabid animals traveled to Paris from across Europe, and even from America and Russia, to receive Pasteur's treatment. Pasteur became a national hero in France, perhaps the most famous scientist in the world. The Pasteur Institute was founded in 1887, funded by international donations, and it remains one of the world's leading biomedical research institutions to this day. But the Pasteur-Koch rivalry was bitter, and it was personal. Part of it was genuinely scientific. Koch criticized Pasteur's anthrax vaccine work, and he had some legitimate points. Pasteur's methods were sometimes less rigorous than Koch's, and Pasteur had a tendency to announce results before all the details were nailed down. Koch published papers pointing out inconsistencies and methodological weaknesses in Pasteur's work, and he wasn't entirely wrong. Pasteur fired back, accusing Koch of narrow-mindedness and technical pedantry, but a lot of it was national. France and Germany had fought a brutal war in 1870 to 1871, the Franco-Prussian War, and France had lost badly. Paris had been besieged. Alsace-Lorraine had been annexed. The wounds were deep, and both Pasteur and Koch carried them. Pasteur was fiercely patriotic and reportedly refused to accept an honorary medical degree from the University of Bonn after the war, returning it with an angry letter. Koch, for his part, published criticisms of Pasteur primarily in German journals. Each man's scientific reputation was a matter of national pride. They sniped at each other through publications, through proxies, through the scientific press of their respective countries. It was ugly at times, and it was petty, but it was also extraordinarily productive, because both men drove each other to higher and higher standards of evidence, pushed relentlessly by the knowledge that the other was watching, waiting, ready to pounce on any error, and in their wake came a flood. The last 2 decades of the 19th century are sometimes called the golden age of microbiology, and for good reason. Koch's methods, his postulates, his staining techniques, his solid culture media, spread through the scientific world like wildfire, and suddenly, one after another, the causative agents of the great infectious diseases were being identified. Koch himself identified the cholera bacillus in 1884, traveling to Egypt and India during cholera epidemics to collect samples. His students and colleagues, armed with his techniques, identified the organisms responsible for an astonishing number of diseases in a remarkably short period. Friedrich Loeffler isolated the diphtheria bacillus in 1884. Georg Gafke cultivated the typhoid bacillus. Shibasaburo Kitasato, a Japanese bacteriologist who had trained in Koch's laboratory, isolated the tetanus bacillus in 1889 and co-discovered the plague bacillus in 1894. Albert Neisser identified the gonococcus. Edwin Klebs and Loeffler together worked out the relationship between diphtheria bacteria and the toxins they produce. Emil von Behring, yet another Koch student, developed antitoxin therapy for diphtheria and won the very first Nobel Prize in Physiology or Medicine in 1901. It was an extraordinary period, a cascade of discovery. Disease after disease, scourge after scourge, traced to specific, identifiable, microscopic organisms. And with identification came the possibility of prevention and treatment on a scale nobody had imagined. But it's worth pausing here to tell a story that happened even before Koch and Pasteur's greatest triumphs, because it shows just how difficult it can be to get people to accept a revolutionary idea, even when the evidence is staring them in the face and lives hang in the balance. In the 1840s, a young Hungarian physician named Ignaz Semmelweis was working at the Vienna General Hospital in the obstetric clinic, and he noticed something horrifying. In the ward where doctors and medical students delivered babies, the death rate from puerperal fever, childbed fever, was appallingly high, sometimes over 10%. 1 in 10 mothers who came to that ward to give birth died of infection shortly afterward. But in the adjacent ward, staffed by midwives, the death rate was far lower, around 2%. The same hospital, the same patients, drastically different outcomes. Semmelweis was tormented by this discrepancy. He investigated every possible explanation: diet, ventilation, laundry practices, birthing positions. Nothing explained it. And then a colleague of his, Jakob Kolechka, died after accidentally cutting his finger during an autopsy, and the pathology of his death looked identical to puerperal fever. And Semmelweis had his terrible insight. The doctors and medical students were going directly from performing autopsies on cadavers to delivering babies without washing their hands. They were carrying something from the dead bodies, some infectious material, on their hands, and introducing it into the mothers during delivery. The midwives, who didn't perform autopsies, didn't carry this contamination. Semmelweis instituted a policy of hand-washing with chlorinated lime solution before examining patients. The death rate in his ward dropped immediately and dramatically, from over 10% to around 1%. The evidence was overwhelming. But here's the tragedy. The medical establishment rejected him. Senior physicians were offended by the suggestion that they, gentlemen and doctors, were carrying death on their hands. Semmelweis was ridiculed, ostracized, eventually dismissed from his position. He grew increasingly erratic and angry, writing furious open letters accusing his colleagues of murder. In 1865, he was committed to a mental asylum, where he was beaten by guards and died 2 weeks later, probably from septicemia, ironically the very type of infection he had spent his career trying to prevent. He was 47 years old. He had been right about everything, and it had destroyed him. Semmelweis's story is one of the most heartbreaking in the history of medicine. He didn't have germ theory to explain why hand-washing worked. He just had the data. And without a theoretical framework, the establishment dismissed the data. It would take Pasteur's germ theory and Lister's antiseptic techniques, arriving years later, to provide the explanation that made hand-washing and surgical cleanliness not just a good idea, but an obvious, inescapable necessity. Joseph Lister, an English surgeon who had been directly inspired by Pasteur's work on germs, introduced antiseptic surgical techniques in the 1860s. Before Lister, surgery was a horrifying gamble. Surgeons operated in their street clothes, sometimes in the same frock coat stiffened with dried blood from hundreds of previous operations. which was considered a badge of experience and seniority, not a sanitation hazard. They didn't wash their hands between patients. They didn't sterilize their instruments. Post-operative infection was so common it was considered a normal, even expected, part of the healing process. Hospital gangrene, septicemia, erysipelas, these were routine killers. Lister changed that. Inspired by Pasteur's demonstration that microbes cause putrefaction, Lister reasoned that wound infection must also be caused by microbes entering the wound from the air or from contaminated hands and instruments. He introduced the use of carbolic acid spray during operations, sterilized surgical instruments, insisted on clean dressings, and developed antiseptic wound treatments. Mortality rates in his surgical wards plummeted. The operating room, once barely distinguishable from a butcher's shop in terms of cleanliness, began its transformation into the sterile environment we know today, and alongside antiseptic surgery came public health infrastructure based on germ theory. Clean water supplies, proper sewage treatment, food safety regulations, quarantine procedures, contact tracing. These unglamorous, infrastructure-level changes, driven by the understanding that specific microbes cause specific diseases and that those microbes can be tracked and controlled, saved more lives in the 20th century than any drug or surgical technique. By some estimates, improvements in sanitation and water treatment alone added more than 20 years to average life expectancy in developed nations. That's the quiet, unglamorous, profound legacy of Pasteur and Koch and Lister and all the rest. But even as scientists were identifying one disease-causing bacterium after another, there was a puzzle lurking at the edges of the field. There were diseases, clearly infectious diseases, that didn't seem to be caused by bacteria at all. You could filter an infectious liquid through a porcelain filter, fine enough to trap every known bacterium, and the liquid that came through, the filtrate, could still cause disease. Something was getting through. Something smaller than any bacterium. Something so small that no microscope of the era could resolve it. And the story of how scientists discovered this something, this entity that sits at the very boundary between living and non-living, begins with tobacco plants and ends with a rethinking of what life itself means. And it starts in the fields and laboratories of late 19th century Europe, where tobacco farmers were losing their crops to a mysterious disease. In the 1870s and 1880s, tobacco growers across the continent were struggling with a condition that devastated their plants. Infected leaves developed a striking mosaic pattern of light and dark green patches, and the disease spread from plant to plant with alarming speed, ruining entire harvests. A German agricultural chemist named Adolf Meyer studied the disease systematically in the 1880s and confirmed that it was infectious. He took sap from diseased plants, rubbed it on healthy ones, and the healthy plants developed the same mosaic pattern. Something in the sap was causing the infection. So far, this followed the standard germ theory playbook. There's an infectious agent. Find it, isolate it, culture it. But Meyer couldn't find any bacterium in the sap. He couldn't culture anything that reproduced the disease. The infectious agent, whatever it was invisible. Then, in 1892, a young Russian botanist named Dmitry Ivanovsky tried the critical experiment. He took infectious sap from diseased tobacco plants and passed it through a Chamberlain filter, one of those unglazed porcelain filters with pores so fine that bacteria could not pass through. This was the definitive test. If the infectious agent was a bacterium, the filter would catch it, and the filtered liquid would be harmless. But the filtered sap was still infectious. Ivanovsky was puzzled but cautious. He suggested that maybe the bacteria were unusually small, or that the filter had a crack in it, or perhaps the bacteria produced a toxin that could pass through the pores. He didn't quite grasp the full significance of what he'd found. It was a Dutch microbiologist named Martinus Beijerinck who, in 1898, repeated Ivanovsky's filtration experiments and took the interpretation to its revolutionary conclusion. Beijerinck confirmed that the filtrate was infectious, and then he went further. He showed that the infectious agent could diffuse through agar gel, which bacteria definitely could not do, suggesting it was something much smaller and perhaps even dissolved in the liquid like a chemical. And when he applied the filtrate to fresh plants, the disease spread vigorously through the new tissue, meaning the agent was multiplying inside the plant. This was not a toxin. A toxin would be diluted with each passage to a new host and eventually lose its potency. Whatever this was, it was increasing in quantity inside living cells. Beijerinck called it a contagium vivum fluidum, a contagious living fluid. He was wrong about the fluid part, as it turned out, but he was right about the essential point. This was a new category of infectious agent, fundamentally different from bacteria, something that had no independent life of its own, but could hijack living cells and multiply within them. He gave it a name. He called it a virus, from the Latin word for poison. And for the next several decades, viruses remained profoundly mysterious. They were too small to see with any light microscope. They couldn't be grown in petri dishes like bacteria, because they only multiplied inside living cells. They passed through filters that trapped everything else. They caused devastating diseases, but they were invisible and unculturable. Scientists knew they existed only by their effects, which was deeply unsatisfying. It was like knowing there's a burglar because your things keep disappearing, but never being able to see the burglar, or even prove the burglar is a physical entity. The breakthrough that shattered this mystery came in 1935, when an American biochemist named Wendell Stanley, working at the Rockefeller Institute, did something that astonished the scientific world. He took the tobacco mosaic virus and crystallized it, like table salt, like sugar. He purified it from ton after ton of infected tobacco juice and produced actual needle-shaped crystals that you could hold in your hand, store on a shelf, weigh on a balance. And those crystals, when dissolved in water and applied to healthy tobacco plants, caused the mosaic disease just as powerfully as fresh infectious sap. A crystal that causes disease, a chemical that reproduces. Think about how profoundly strange that is. Crystals aren't alive. They don't eat, they don't breathe, they don't metabolize. They're just molecules arranged in a repeating geometric lattice. But these crystals were infectious. They could multiply, but only inside a living cell. They existed in a twilight zone between chemistry and biology that nobody had imagined possible. It wasn't until the invention of the electron microscope in the late 1930s and its refinement through the 1940s that scientists could finally see what viruses actually looked like. And what they saw was remarkable. Viruses came in an incredible variety of forms. Some were roughly spherical, like tiny soccer balls with geometric facets. Some were rod-shaped, some were bullet-shaped, and some, the bacteriophages that infect bacteria, looked like nothing so much as tiny lunar landers, with icosahedral heads perched on spindly tail fibers. The tobacco mosaic virus itself turned out to be a hollow rod about 300 nanometers long, made of over 2,000 identical protein subunits, arranged in a tight helix, with a single strand of RNA coiled inside like a scroll in a tube. And that basic architecture, a coat of protein surrounding a core of nucleic acid, DNA or RNA, turned out to be the fundamental plan of all viruses, from the simplest to the most complex. They are, at their most essential, a set of genetic instructions wrapped in a protein shell. Some of them, like influenza and HIV, also have a lipid envelope stolen from their previous host cell, but the core concept is the same: no metabolism, no ability to reproduce on their own, no cellular machinery. They hijack the machinery of living cells, inject their genetic material, commandeer the cell's ribosomes and enzymes, force the cell to produce thousands of copies of the virus, and then, typically, the cell bursts open and dies, releasing a flood of new virus particles to infect new cells. It's parasitism at the molecular level, elegant and ruthless. Are viruses alive? That question has been debated for over a century, and there's still no real consensus. They have genetic material. They evolve through mutation and natural selection. They have complex, sophisticated structures, but they can't reproduce without a host. They have no metabolism of their own. They can be crystallized and stored on a shelf indefinitely, inert as a grain of sand, and spring back into infectious action the moment they contact a suitable cell. Most biologists today think of them as obligate intracellular parasites, biological entities that are functionally part of the living world, even if they don't tick every box on the traditional checklist for life. But the question itself reveals something important, which is that our definition of life might be too narrow, too tied to the particular way cells work, to capture everything that exists on the spectrum between inert chemistry and full-blown biology. And whatever their metaphysical status, viruses have shaped human history to a degree that's almost impossible to overstate. Smallpox alone killed an estimated 300 million people in the 20th century, before it was eradicated through vaccination in 1980. The influenza pandemic of 1918, caused by an H1N1 virus, killed somewhere between 50 and 100 million people worldwide, more than all the soldiers killed in the First World War, in just about 2 years. It swept across the planet in 3 waves, the second wave in the autumn of 1918 being by far the deadliest. Cities were overwhelmed. Morgues overflowed. In Philadelphia, which held a massive Liberty Loan parade in September 1918, despite warnings from public health officials, the death toll was catastrophic. Within days of the parade, every bed in every hospital in the city was full. Within weeks, over 12,000 Philadelphians were dead. Bodies were stacked in homes and on porches because there weren't enough coffins, enough undertakers, enough gravediggers to handle the dead. What made the 1918 flu so terrifying, beyond its sheer scale, was its predilection for killing healthy young adults, a pattern almost opposite to typical influenza, which is most dangerous to the very young and the very old. The best current theory is that the virus triggered a cytokine storm, an overwhelming immune overreaction, and that the strongest immune systems, those of young, healthy adults, produced the most violent and self-destructive responses. Your own immune system, essentially, was killing you. It was a grim illustration of a theme that runs through all of microbiology, the intimate, sometimes deadly interplay between the microbial world and the systems we've evolved to deal with it. And then there was polio. The story of polio and its vaccine is one that's worth slowing down for, because it captures something essential about the intersection of science, public fear, and the kind of focused, determined effort that can achieve things that once seemed impossible. In the first half of the 20th century, polio was one of the most feared diseases in the world, especially in the United States. Every summer, parents dreaded the arrival of polio season. Swimming pools closed, movie theaters were empty, children were kept indoors. The disease struck seemingly at random, mostly affecting children, paralyzing limbs and, in the most severe cases, paralyzing the muscles needed to breathe. The image of rows of children in iron lungs, those coffin-like mechanical ventilators that breathed for patients whose chest muscles had been destroyed, became one of the most haunting images of American medicine. The most famous polio patient was Franklin D. Roosevelt, who contracted the disease in 1921 at the age of 39. Roosevelt was left permanently paralyzed from the waist down, though he went to extraordinary lengths to conceal the extent of his disability from the public, rarely being photographed in his wheelchair, using leg braces and the arms of aides to stand at podiums. He founded the National Foundation for Infantile Paralysis, which became known as the March of Dimes, and which funded the research that would ultimately defeat the disease. Jonas Salk was the scientist who developed the first successful polio vaccine. A microbiologist from New York City, the son of Russian Jewish immigrants who had never attended high school themselves, Salk had trained in virology and spent years working on influenza vaccines before turning his attention to polio. His approach was to use a killed virus vaccine. He grew poliovirus in monkey kidney cell cultures, killed it with formaldehyde so it could no longer replicate, and injected the inactivated virus. The dead virus couldn't cause disease, but its proteins could still be recognized by the immune system, stimulating the production of protective antibodies. Salk tested the vaccine first on himself, his wife, and his 3 sons. Then, in 1954, came the largest medical experiment in American history up to that point. Nearly 2 million children across the United States participated in the field trial. Some received the vaccine, some received a placebo, in a carefully designed double-blind study. The results were announced on April 12, 1955, the 10th anniversary of Roosevelt's death, and they were triumphant. The vaccine was 80 to 90% effective against paralytic polio. It was safe. It worked. Church bells rang in cities across America. People wept in the streets. Salk became an overnight national hero. When asked who owned the patent on the vaccine, he gave a reply that has echoed through the decades. The people. Could you patent the sun? He never patented it. He never profited from it personally. Albert Sabin later developed an oral polio vaccine, using a live but attenuated form of the virus, which had important advantages for mass vaccination campaigns, particularly in developing countries, because it could be administered by mouth rather than injection, and could spread through communities, immunizing even people who hadn't been directly vaccinated. Together, the Salk and Sabin vaccines drove polio to the brink of global eradication. In 1988, there were an estimated 350,000 cases of polio worldwide. By 2023, that number had dropped to handfuls, with cases persisting only in a few regions of Afghanistan and Pakistan. It's one of the greatest public health achievements in human history, and it was accomplished by understanding how a virus works and engineering a way to teach the immune system to fight it before the infection arrives. But while virologists were grappling with the smallest and most enigmatic infectious agents, another revolution was quietly gathering force in microbiology, one that would transform not just our understanding of microbes, but our understanding of life itself. And it started with an experiment that didn't seem all that impressive at the time, carried out by a quiet, self-effacing researcher who never received the Nobel Prize he almost certainly deserved. His name was Oswald Avery, and what he showed, working at the Rockefeller Institute in New York in the early 1940s, would change everything we know about how living things store, transmit, and use genetic information, because Avery demonstrated, using bacteria, that the molecule carrying hereditary information, the substance of genes themselves, is DNA, deoxyribonucleic acid. And this discovery, made with humble pneumococcus bacteria in a Manhattan laboratory, laid the foundation for the entire molecular revolution in biology that followed. Here's the story. Back in 1928, a British bacteriologist named Frederick Griffith had performed a peculiar and unsettling experiment with Streptococcus pneumoniae, the bacteria that cause certain forms of pneumonia. There were 2 strains. One was virulent, a killer, and it had a smooth, glistening appearance when grown on agar plates, because each cell was surrounded by a thick polysaccharide capsule that shielded it from the immune system. The other strain was harmless, rough-looking on plates, because it lacked the capsule. If you injected mice with the smooth strain, they died of pneumonia within days. If you injected them with the rough strain, they shrugged it off. Then Griffith did something unexpected. He took the smooth, virulent strain and killed it with heat. Dead cells, completely dead, no possibility of reproducing. He injected these dead, smooth bacteria into mice along with the living, rough, harmless strain. Logically, the mice should have been perfectly fine. The smooth cells were dead, the rough cells were harmless. 2 harmless things combined should still be harmless. But the mice died. And when Griffith examined their blood, he found living, smooth bacteria, complete with capsules, reproducing and killing, as efficiently as if they'd never been heat-treated at all. Something from the dead, smooth cells had been taken up by the living, rough cells and had permanently transformed them, converting them from harmless to deadly. And this transformation was heritable. The descendants of the transformed cells were also smooth and virulent. Whatever had passed between the cells carried genetic information. Griffith called this phenomenon transformation, and it created a sensation. But the burning question was obvious. What was the transforming substance? What molecule carried the genetic information from the dead cells to the living ones? Most scientists assumed it was protein. Proteins were the most complex and versatile molecules known. DNA had been discovered decades earlier by Friedrich Miescher, who isolated it from white blood cells in 1869, but it was widely considered a boring, structurally repetitive molecule, too simple to carry the enormous complexity of genetic instructions. Surely the genes, whatever they were, must be made of protein. Avery and his colleagues, Colin MacLeod and Macklin McCarty, spent over a decade working on this problem. It was painstaking, meticulous, unglamorous work. They systematically purified the transforming substance, stripping away layer after layer, protein, lipid, carbohydrate, RNA, testing at each step whether the substance could still transform rough cells into smooth ones. They treated it with proteases, enzymes that destroy proteins. The transforming activity survived. They treated it with ribonuclease, which destroys RNA, still active. They treated it with deoxyribonuclease, an enzyme that specifically destroys DNA. The transforming activity vanished. The transforming principle was DNA. Avery published these results in 1944, and they should have set the scientific world on fire. But the reception was surprisingly muted. Many prominent scientists, including some of the leading geneticists of the time, remained skeptical. They questioned the purity of Avery's DNA preparations, suggested that trace protein contamination might be responsible, resisted the implication that DNA, this seemingly simple molecule, could carry the complexity of genetic information. Part of the resistance was scientific caution. Part of it was the weight of received wisdom, the decades-long assumption that proteins were the molecules of heredity. Avery himself was modest and cautious in his claims, never pushing his interpretation as aggressively as he might have. He never received the Nobel Prize, and many molecular biologists consider this one of the greatest oversights in the history of the prize. The definitive confirmation came in 1952 from Alfred Hershey and Martha Chase, who used bacteriophages, those viruses that infect bacteria, to settle the question once and for all. They radioactively labeled the protein coat of the phage with one isotope and the DNA core with another, then let the phages infect bacteria. The DNA entered the bacterial cells and directed the production of new phages. The protein stayed outside. Genes were made of DNA. The case was closed. And then, the following year, came the double helix. In 1953, James Watson and Francis Crick, working at the Cavendish Laboratory in Cambridge, England, proposed the three-dimensional structure of DNA. Two polynucleotide strands wound around each other in a right-handed helix, the sugar-phosphate backbones on the outside, the nitrogenous bases on the inside, hydrogen bonded to each other in specific complementary pairs, adenine with thymine, guanine with cytosine. The structure was breathtaking in its elegance and immediately suggested how DNA could fulfill its biological roles. Replication. Unwind the two strands, and each serves as a template for a new complementary strand. Information storage. The sequence of bases along the strand, like letters in an enormous molecular alphabet, could encode any message of any length and any complexity. Now, the Watson and Crick story has been told many times, and it carries a shadow that should not be glossed over. Much of their crucial structural insight depended on X-ray crystallography data produced by Rosalind Franklin and her student Raymond Gosling at King's College London. Franklin was a brilliant experimental physicist who had generated the finest X-ray diffraction images of DNA in the world. Her photo 51, taken in May 1952, clearly showed the helical structure and provided key measurements of the dimensions and symmetry of the molecule. Watson and Crick saw this data without Franklin's explicit knowledge or permission. It was shown to them by Maurice Wilkins, Franklin's colleague at King's, with whom she had a famously difficult working relationship. Watson later described seeing photo 51 as a pivotal moment, and Crick himself acknowledged that Franklin's data was essential to their model. Franklin died of ovarian cancer in 1958, at the age of 37, possibly from the cumulative effects of radiation exposure during her X-ray work. When Watson, Crick, and Wilkins received the Nobel Prize for the DNA structure in 1962, Franklin was not honored. Nobel Prizes are not awarded posthumously, and there is an ongoing and justified debate about whether she received adequate credit during her lifetime and in the years that followed. Watson's memoir, published in 1968, portrayed her in ways that many scientists and historians have found dismissive and unfair. It's a complicated story without clean heroes and villains, but what is not complicated is this. Franklin's contribution to the discovery of the DNA structure was essential, and the history of molecular biology would be poorer and less honest without acknowledging it fully. But the structure of DNA, however it was discovered, opened the floodgates. Over the next 2 decades, a torrent of discoveries poured out. The genetic code was deciphered, largely through the work of Marshall Nierenberg, Har Gobind Khorana, and others, revealing that sequences of 3 DNA bases, called codons, each specify a particular amino acid in a protein. The code turned out to be essentially universal. The same codons specify the same amino acids in bacteria, in humans, in plants, in mushrooms, in virtually every living thing on Earth. This universality is one of the most powerful pieces of evidence for the common ancestry of all life. From the simplest bacterium to the most complex animal, we all read the same genetic language. We're all reading from the same book, written in the same alphabet, using the same rules of grammar. The differences between a bacterium and a blue whale are differences of content, not of code. And here's where microbiology at the molecular level gets genuinely wild. Because bacteria, it turns out, are far more genetically dynamic, far more flexible in how they handle their DNA than anyone had imagined. They don't just pass DNA from parent to daughter cell, the way you inherit genes from your parents. They also share DNA horizontally, between completely unrelated cells, across species boundaries, through mechanisms that have no real equivalent in the animal world. One of these mechanisms is called conjugation. A donor bacterium extends a thin, tubular appendage, called a pilus, toward a recipient cell, makes physical contact, and transfers a copy of a piece of its DNA, usually a small circular molecule called a plasmid, directly into the recipient. It's sometimes called bacterial sex, though it's quite different from animal reproduction. There's no fusion of cells, no mixing of chromosomes. It's more like one cell handing another a flash drive. Another mechanism is transformation, the very process Griffith discovered back in 1928, where bacteria pick up free-floating DNA from their environment, fragments released from dead or ruptured cells, and incorporate those foreign genes into their own genome. And a third is transduction, where bacteriophages accidentally package a piece of bacterial DNA instead of their own viral DNA and ferry it to a new cell when they infect it, delivering someone else's genes by viral courier. This horizontal gene transfer, as it's called, is one of the most important concepts in all of modern biology, and it has enormous, terrifying practical consequences. Because it means that useful genes, including genes that confer resistance to antibiotics, can spread not just from parent to offspring, but sideways, between completely different species of bacteria, in a single event. A gene that gives one species of gut bacterium the ability to produce an enzyme that destroys penicillin can be passed, on a plasmid, to a completely different species, one that had never encountered penicillin in its evolutionary history, in a matter of hours. And this is exactly what has been happening, on a massive, accelerating scale, since the introduction of antibiotics in the 1940s. And this brings us to one of the most important and sobering stories in all of microbiology, the story of antibiotics, from miracle to crisis. When Alexander Fleming noticed in 1928 that a mold growing on a petri dish in his laboratory at St. Mary's Hospital in London had killed the surrounding staphylococcal bacteria, he stumbled onto one of the most important discoveries in medical history. The mold was Penicillium notatum, and the bacteria-killing substance it produced would be named penicillin. But Fleming couldn't purify enough of the substance to be medically useful, and the discovery sat largely dormant for over a decade. It was Howard Florey and Ernst Boris Chain, working at Oxford during the darkest days of World War II, who turned penicillin from a laboratory curiosity into a life-saving drug. The challenges were enormous. Producing penicillin in quantity required growing vast amounts of the mold and extracting the active compound, a process that was initially so inefficient that the researchers famously collected the urine of treated patients to recover and recycle the precious drug. Eventually, mass production was scaled up with American industrial help, and by D-Day in 1944, there was enough penicillin to treat every wounded Allied soldier. Infections that had been death sentences, pneumonia, septicemia, gangrene, could suddenly be cured. The antibiotic era had begun, and it felt like a miracle. But Fleming himself saw the danger. In his Nobel lecture in 1945, he issued a prophetic warning. He cautioned that improper use of penicillin, using too little of it, or using it for trivial conditions, could encourage the development of resistant bacteria. The time may come, he said, when penicillin can be bought by anyone in the shops. Then there is the danger that the ignorant man may easily underdose himself and, by exposing his microbes to non-lethal quantities of the drug, make them resistant. He was right, and it happened faster than almost anyone expected. Within a few years of penicillin's widespread use, resistant Staphylococcus strains appeared. Methicillin was developed specifically to combat penicillin-resistant staph, and then methicillin-resistant Staphylococcus aureus, Messer SA, emerged. New antibiotics were developed, and resistance to each one followed, sometimes within a few years. It's an arms race, and the bacteria have the advantage of numbers and speed. A single bacterial cell can divide every 20 minutes. In a day, One cell can become billions. Each division is a chance for a mutation, and through horizontal gene transfer, a single resistance gene can spread through an entire bacterial community, even across species barriers, in days or weeks. Today, antibiotic resistance is one of the most serious threats to global public health. Drug-resistant infections already kill well over 1 million people per year worldwide. There are strains of tuberculosis resistant to virtually every antibiotic available, what's called extensively drug-resistant TB. There are strains of gonorrhea approaching untreatable status. We face the real possibility of a post-antibiotic era, a world where routine surgeries become dangerous again, where a scraped knee could lead to a fatal infection, where we lose the medical advances that have saved hundreds of millions of lives over the last 80 years. And the tragedy is that much of this crisis is self-inflicted. Overuse of antibiotics in human medicine, prescribed for viral infections like colds and flu, that antibiotics can't treat. Massive use in agriculture, where antibiotics are fed to healthy livestock as growth promoters, a practice that creates enormous reservoirs of resistant bacteria. Every unnecessary dose is a selection pressure, favoring the survival and reproduction of resistant organisms and eliminating the susceptible ones. We are breeding superbugs through our own negligence. But the story of microbiology isn't only about diseases and the microbes that cause them. It's also about the staggering diversity of microbial life, a diversity so vast that we're only beginning to appreciate it. And the person who forced us to reckon with that diversity, who literally redrew the tree of life to account for it, was a microbiologist named Carl Woese, working at the University of Illinois at Urbana-Champaign, using a molecule called ribosomal RNA as his compass. Here's the problem Woese was trying to solve. For most of the 20th century, biologists classified microbes by what they looked like under a microscope and how they behaved in lab tests. What shape are they? What sugars can they ferment? What temperatures can they tolerate? What stains do they absorb? But these were crude, often misleading measures. Two bacteria that looked identical under a microscope might be as genetically different from each other as a human is from a mushroom, and the vast, vast majority of microbial species couldn't be cultured in the laboratory at all. They existed in soil, ocean sediments, hot springs, deep rock, the guts of termites, the surfaces of deep-sea volcanic vents, environments that were incredibly difficult or impossible to replicate in a petri dish. Even today, optimistic estimates suggest we can culture less than 1% of all microbial species on Earth. Less than 1%. The other 99% are the microbial dark matter of the biosphere, invisible to traditional methods. Woese's breakthrough was to use a molecular yardstick instead. Ribosomes, the cellular machines that translate genetic information into proteins, exist in every living cell. And a key component of ribosomes, the 16S ribosomal RNA gene in bacteria and archaea, evolves slowly enough to serve as a molecular clock. Compare the 16S RNA sequences of two organisms. If the sequences are very similar, the organisms diverged recently. If the sequences are very different, they diverged a long time ago. By building family trees based on these molecular comparisons, Woese could map the relationships between organisms that looked nothing alike and uncover deep evolutionary connections that morphology alone could never reveal. In the late 1970s, Woese and his colleague George Fox published findings that shook biology to its foundations. They had been systematically sequencing ribosomal RNA from every microbe they could obtain, and they found something nobody expected: a whole group of microorganisms, organisms that had been classified as bacteria because they looked like bacteria, functioned like bacteria, were roughly the same size as bacteria, turned out to be as genetically different from true bacteria as bacteria are from animals and plants. They were a separate domain of life, a third major branch on the tree of life that nobody had recognized. Woese called them archaebacteria, later shortened to archaea. The scientific establishment was not kind to Woese. Many prominent biologists dismissed his findings. Some attacked his methods. Others attacked him personally. The great microbiologist Salvador Luria reportedly called the work a joke. The resistance was intense, and some of it was ugly. Woese spent years defending his work against skeptics who simply couldn't accept that the tree of life had a major branch that everyone had overlooked. But the data was overwhelming, and over time, the scientific community came around. Today, the three-domain system, bacteria, archaea, and eukarya, is universally accepted. Carl Woese redrew the tree of life, and he was right. And the archaea turned out to be endlessly fascinating in their own right. Many of the first archaea to be studied were extremophiles, organisms thriving in environments that would be lethal to virtually any other form of life. There are thermophiles, growing in hot springs at temperatures above 100 degrees Celsius, in water so hot it would scald you in seconds. Pyrolobus fumarii, discovered in a deep-sea hydrothermal vent, can grow at 113 degrees Celsius. There are halophiles living in salt lakes so concentrated that crystals of salt form on their cell surfaces, in environments like the Dead Sea and the Great Salt Lake. There are methanogens, archaea, that produce methane gas as a metabolic byproduct, living in the oxygen-free depths of swamps, in landfills, in the digestive tracts of cows and termites. There are acidophiles thriving in drainage from mines at a pH of 0 or 1, conditions as acidic as battery acid. There are psychrophiles growing in Antarctic ice. There are organisms living kilometers beneath the Earth's surface, in solid rock, surviving on chemical energy, with no access to sunlight whatsoever. These extremophiles expanded our understanding of where life can exist in ways that have profound implications. Think about the search for life beyond Earth. If microbes on our own planet can thrive in boiling acid, in frigid ice, in solid rock kilometers underground, in superheated water around volcanic vents where no sunlight has ever penetrated, then the range of environments where extraterrestrial life might exist is vastly larger than anyone had imagined a few decades ago. Jupiter's moon Europa, with its subsurface ocean of liquid water heated by tidal forces, covered by a shell of ice, is now considered one of the most promising places to look for extraterrestrial life. Saturn's moon Enceladus, which shoots geysers of water vapor into space from cracks in its icy surface, is another candidate. Even Mars, cold and dry and bathed in radiation as it is, might harbor microbial life deep underground, surviving on chemical energy in the Martian rock, the way deep subsurface organisms survive on Earth. And here's something worth thinking about, maybe the most profound thing in all of microbiology. Microbes aren't just passengers on this planet. They didn't just show up and find a world ready-made for them. They made it. They shaped the Earth, transformed its atmosphere, sculpted its chemistry, and created the conditions that allowed everything else, including you, to exist. For the first 2 billion years of life's history, from roughly 3.5 billion years ago to about 1.5 billion years ago, microbes were the only life on Earth. There were no plants, no animals, no fungi, no multicellular organisms of any kind, just microbes, bacteria and archaea, in the oceans, in the sediments, in the hot springs, in every environment the planet offered. And during those 2 billion years, they did something extraordinary. a group of photosynthetic bacteria, the cyanobacteria, sometimes misleadingly called blue-green algae, even though they're not algae at all, invented oxygenic photosynthesis. They figured out how to use sunlight to split water molecules and release oxygen as a waste product. Think about that. A metabolic waste product of ancient bacteria became the gas that you're breathing right now. At first, the oxygen they produced didn't accumulate in the atmosphere. It was soaked up by dissolved iron in the oceans, reacting with it to form iron oxides that settled to the seafloor in great rusty layers. These are the banded iron formations that geologists find all over the world. Ancient sedimentary rocks, striped with bands of iron-rich and iron-poor minerals. And they're essentially a geological record of cyanobacteria, slowly pumping oxygen into the oceans over hundreds of millions of years. You can hold a piece of banded iron formation in your hand, a rock 2.5 billion years old, and you're holding the literal byproduct of microbial photosynthesis. Eventually, the oceanic iron sinks were saturated. There was no more dissolved iron to absorb the oxygen, and the oxygen began to build up in the atmosphere. This event, called the Great Oxidation Event, happened roughly 2.4 billion years ago, and it was almost certainly the most consequential event in the history of life on Earth, because oxygen is, from the perspective of anaerobic organisms, a deadly poison. It's a highly reactive molecule that rips apart the biochemistry of organisms not equipped to handle it. The rise of atmospheric oxygen was, for the anaerobic microbes that dominated the early Earth, a catastrophe of unimaginable proportions. A mass extinction event that may have wiped out a significant fraction of all life on the planet. The cyanobacteria had, in effect, polluted their own world with their own waste product, and the casualties were enormous. But that catastrophe created the opportunity for something new. Organisms that could tolerate oxygen, and eventually organisms that could use oxygen as a fuel for metabolism, aerobic respiration, had an enormous energetic advantage. Aerobic metabolism extracts far more energy from food molecules than anaerobic fermentation does, roughly 18 times more per molecule of glucose. And that extra energy, made available by the oxygen that cyanobacteria had pumped into the air, eventually powered the evolution of complex multicellular life, of animals and plants and fungi and everything else that came after. Your ability to breathe, to run, to think, to lie here in the dark listening to this, is a direct consequence of what cyanobacteria did 2.5 billion years ago. You are running on ancient bacterial exhaust, and the nitrogen in your proteins was fixed from the atmosphere by nitrogen-fixing bacteria. The soil in which your food grows was built by microbial communities over millennia. The carbon cycle, the nitrogen cycle, the sulfur cycle, the phosphorus cycle, all the great biogeochemical cycles that keep the Earth habitable, are driven primarily by microbes. If all microbes on Earth vanished tomorrow, complex life would collapse within weeks. The cycles would stop. Nutrients would stop being recycled. Dead organic matter would stop being decomposed. The oceans would stagnate. The atmosphere would gradually change. Within a relatively short time, geologically speaking, the Earth would become uninhabitable for most of the life that currently exists. But if all complex life vanished, if every animal and plant and fungus disappeared, the microbes would carry on just fine. They'd barely notice. They were here for 2 billion years before complex life existed, and they'd be fine without it. We need them. They don't need us. So when you hear the word microbe and think of something small, something trivial, something to be washed off your hands with soap, remember that. Microbes are the most successful, most durable, most ecologically important forms of life on this planet. They have been running the show for 3.5 billion years, and they will continue to run it long after humans, and everything we've built are fossils in the rock. But you don't have to go to space or deep into geological time to find astonishing microbial diversity. You can find it much closer to home, inside yourself, in fact. The human microbiome, the sum total of all the microorganisms living in and on your body, has become one of the most exciting and fastest-growing areas of research in all of biology. The Human Microbiome Project, launched by the National Institutes of Health in 2007, set out to characterize the microbial communities in the human body, and the picture that emerged was extraordinary. Your gut alone harbors hundreds of different species of bacteria, collectively possessing millions of genes, far more genetic diversity than the roughly 20,000 protein-coding genes in your own human genome. These gut bacteria perform an astonishing range of functions. They break down complex carbohydrates, dietary fibers, and plant polysaccharides that your own enzymes cannot digest, producing short-chain fatty acids like butyrate that nourish the cells lining your intestine and help maintain the integrity of the gut barrier. They synthesize vitamins, including vitamin K and several B vitamins. They metabolize drugs, hormones, and dietary compounds, sometimes activating them, sometimes inactivating them. They compete with potential pathogens for space and nutrients, forming a defensive ecological barrier. And they communicate with your immune system in complex ways, helping to calibrate the balance between immune activation and tolerance, teaching the immune system to distinguish between harmless commensal organisms and genuine threats. And there's growing evidence, still being fleshed out but increasingly compelling, that the gut microbiome influences far more than digestion. Studies have linked the composition of gut bacterial communities to obesity, type 2 diabetes, cardiovascular disease, inflammatory bowel disease, autoimmune conditions, allergies, and even mental health. The gut-brain axis, the bidirectional communication network linking the gut microbiome to the central nervous system, is a rapidly expanding field. Gut bacteria produce neurotransmitters, including serotonin, dopamine, and gamma-aminobutyric acid, or GABA. They influence the vagus nerve, the major neural highway connecting the gut to the brain. Animal studies have shown that germ-free mice, raised in sterile conditions with no gut bacteria at all, display altered behavior, including increased anxiety and abnormal stress responses, and that these changes can be partially reversed by introducing specific bacterial species. Now, a word of caution. The microbiome field is exciting, genuinely exciting, but it's also young, and some of the headlines that appear in popular media run ahead of the evidence. Many of the associations between specific microbial compositions and specific health outcomes are correlational, not causal. The human microbiome is extraordinarily complex, varying enormously from person to person, influenced by diet, geography, genetics, age, medication use, mode of birth delivery, and dozens of other factors. Simple stories about single species causing single conditions are rarely accurate, but the broad picture is becoming clear, even if the details remain fuzzy. We are not solitary organisms. We are ecosystems, and the health of our microbial ecosystem matters, probably a great deal, to our overall well-being. This ecological understanding represents a genuinely profound shift in how we think about health and disease. For more than a century, since Pasteur and Koch, the dominant metaphor in medicine has been military. Microbes are invaders. The body is a fortress. Antibiotics are weapons. Kill the germs, cure the disease. And that metaphor has been enormously productive. It saved countless millions of lives. But the microbiome revolution reveals it as incomplete. Yes, some microbes are enemies that need to be fought. But the vast majority of the microbes in your body are allies, or at least neutral neighbors, and indiscriminate warfare against them, through broad-spectrum antibiotics, through excessive sanitization, through the sterile, hyper-clean environments of modern life, can do real damage. Disrupting the gut microbiome with antibiotics can open the door to opportunistic infections like Clostridioides difficile, which can colonize the gut when antibiotic treatment eliminates the normal protective flora, causing severe, sometimes life-threatening diarrhea. The future of medicine may look less like warfare and more like gardening, tending microbial communities, cultivating beneficial species, restoring ecological balance when it's disrupted. Probiotics, prebiotics, fecal microbiota transplantation, precision antibiotics that target specific pathogens without harming commensals. All of these approaches reflect a shift toward ecological thinking in clinical practice. And, in a beautiful historical irony, some of the most promising new approaches to bacterial infection involve reaching back to a therapy that predates antibiotics entirely. Phage therapy, the use of bacteriophages to treat bacterial infections, was first explored in the 1910s and 1920s by Félix Derel, a colorful, self-taught French-Canadian microbiologist who co-discovered bacteriophages and immediately grasped their therapeutic potential. Derel was a remarkable character, largely self-educated, perpetually restless, the kind of person who taught himself microbiology from library books and then traveled the world applying what he'd learned. He'd worked on locust plagues in Argentina, cholera outbreaks in Indochina, dysentery in France. While studying dysentery at the Pasteur Institute in Paris during World War I, he noticed something strange. When he filtered stool samples from recovering soldiers through porcelain filters fine enough to remove all bacteria, and then added the filtrate to fresh bacterial cultures, the bacteria were killed. Something invisible was destroying them. He called these invisible agents bacteriophages, eaters of bacteria, and by 1919, he was using them to treat patients. Some of the early results were striking. Children with severe dysentery recovered rapidly after a single dose of phage preparation. Derel traveled to India, Egypt, and the Soviet Union, promoting phage therapy wherever he went. But the arrival of antibiotics in the 1940s pushed phage therapy to the margins in Western medicine. Antibiotics were easier to mass-produce, simpler to prescribe, and their broad spectrum of action seemed like an advantage at the time. Phage therapy continued to be practiced at institutions like the Eliava Institute in Tbilisi, Georgia, throughout the Soviet era, but it was largely forgotten in the West. Now, with antibiotic resistance growing, researchers around the world are revisiting phage therapy with modern tools, engineering phages with precision, characterizing phage bacteria interactions at the molecular level, conducting clinical trials. In 2016, Tom Patterson, a professor at the University of California, San Diego, was near death from a multi-drug-resistant Acinetobacter baumannii infection that no antibiotic could touch. His wife, an infectious disease epidemiologist, helped coordinate an emergency effort to find phages that could attack his specific strain. They worked. Patterson recovered. It was one of the first well-documented cases of intravenous phage therapy, saving a life in the modern era, and it helped reignite interest in the field. Phage therapy isn't a magic bullet. It has its own challenges. Bacteria can evolve resistance to phages too, but it's a promising addition to the arsenal, and it's deeply satisfying that a century-old idea is finding new relevance in the age of molecular biology. And speaking of molecular tools, there is one discovery from the microbial world that has transformed all of biology so profoundly, so rapidly, that it's worth spending a few quiet moments on, even as you're sinking deeper into your pillow. It's called CRISPR, and the beautiful thing about it is that it was hiding in bacteria all along, for billions of years. We just didn't know what we were looking at. CRISPR stands for clustered, regularly interspaced, short palindromic repeats, which is possibly the least poetic name ever given to one of the most elegant discoveries in the history of science. But here's what it actually is. It's a bacterial immune system. Bacteria have been fighting viruses for as long as both have existed, billions of years. And bacteria evolved a defense. When a bacterium survives a viral attack, it can snip out a small piece of the invading virus's DNA and store it in a special region of its own genome, the CRISPR array. These stored sequences function like a molecular mugshot file, a library of past invaders. If the same virus, or a close relative, attacks again, the bacterium can recognize the incoming viral DNA by matching it against the stored sequences, and then it deploys a protein called Cas9, which acts like a pair of molecular scissors, cutting the viral DNA at a precise location and destroying it. Scientists had noticed these odd repetitive sequences in bacterial genomes since the late 1980s, beginning with work by Yoshizumi Ishino and colleagues in Japan, who spotted them in E. coli but had no idea what they were for. Over the next 2 decades, researchers including Francisco Mojica in Spain, Ruud Janssen in the Netherlands, Alexander Bolotin in France, and Philippe Horvath and Rodolphe Barangot, working with yogurt cultures, gradually pieced together the puzzle. Mojica, in particular, deserves enormous credit. Working in relative obscurity at the University of Alicante, he was the one who recognized that the spacer sequences between the repeats matched sequences from known bacteriophages and other mobile genetic elements. He realized, around 2003, that CRISPR was an adaptive immune system. Getting this insight published took years of rejection from top journals, a familiar story in science. And then, in 2012, Jennifer Doudna at UC Berkeley and Emmanuel Charpentier, then at Umeå University in Sweden, published the landmark paper that ignited a revolution. They showed that the CRISPR-Cas9 system could be reprogrammed. You could design a synthetic guide RNA that would direct the Cas9 protein to cut any DNA sequence you chose, in any organism, with extraordinary precision. It was a programmable genetic scalpel, simple to use, cheap to produce, and astonishingly effective. You could edit the genome of any living thing with a precision that would have been unthinkable just a few years earlier. The implications were immediate and staggering. Within months of the paper's publication, laboratories around the world were using CRISPR to edit genes in mice, fruit flies, zebrafish, rice, wheat, human cells, and culture. Experiments that would previously have taken years and cost millions of dollars could now be done in weeks for a few hundred. Doudna and Charpentier were awarded the Nobel Prize in Chemistry in 2020, the first Nobel ever awarded to 2 women alone, without a male collaborator. The applications continue to multiply. CRISPR-based therapies are moving through clinical trials for genetic diseases like sickle cell anemia and beta thalassemia. In December 2023, the first CRISPR-based therapy, Casgevy, was approved for clinical use in the UK and the United States. Agricultural applications include crops engineered for drought resistance, disease resistance, and improved nutrition. CRISPR-based diagnostic tools can detect specific viral or bacterial sequences with extraordinary sensitivity, and the technology keeps improving, with new variants like base editors and prime editors offering even greater precision and versatility. And here's the thing I find most beautiful about this entire story. The most powerful tool in modern biotechnology was discovered by studying the immune systems of bacteria, organisms we couldn't even see until 350 years ago. Organisms that many people casually dismiss as simple, primitive, even disgusting. But bacteria have been evolving for 3.5 billion years. They have been locked in an arms race with viruses for just as long. They have developed molecular defenses of exquisite sophistication, and by studying those defenses with patience and curiosity, we unlocked a tool that could change the course of human medicine, agriculture, and biotechnology forever. The lesson is as old as Leeuwenhoek's lenses. Look closely enough at the microbial world, and you'll find things you never imagined. And CRISPR is just one facet of a broader revolution that's unfolding right now. One that's turning microbiology from a science of observation into a science of creation. Synthetic biology, the discipline of designing and building biological systems that don't exist in nature, draws heavily on our understanding of microbial genetics and metabolism. Researchers are engineering bacteria to produce biofuels, to manufacture pharmaceuticals, to detect environmental pollutants, to break down plastic waste. In 2010, Craig Venter's team created the first synthetic cell, a bacterial cell whose entire genome had been designed on a computer and assembled in the laboratory from chemical building blocks. They named it Cynthia, and while it was essentially a copy of an existing bacterial species rather than a truly novel organism, it demonstrated something remarkable. The genetic software of a living cell could be written from scratch, booted up in a cellular chassis, and brought to life. The boundary between the natural and the artificial in the microbial world is blurring. There are also researchers engineering bacteria to serve as living medicines, organisms designed to colonize the gut and produce therapeutic molecules on site, or to seek out and destroy tumor cells from the inside. Others are designing microbial communities from the ground up, synthetic ecosystems of multiple engineered species that work together to accomplish tasks no single organism could manage alone. It sounds like science fiction, and a few years ago it would have been. But the tools are here, and the pace of progress is extraordinary. The pandemic we all lived through not so long ago was a vivid, painful, globe-spanning reminder of how much the microbial world still shapes our lives. SARS-CoV-2, the coronavirus that caused COVID-19, emerged in late 2019 and swept across the planet in weeks, killing millions, overwhelming health systems, shutting down economies, and transforming daily life in ways that would have seemed unthinkable just months before. And the tools we use to fight it, every single one, were products of microbiology. PCR testing, invented by Kary Mullis in 1983, originally developed to amplify tiny quantities of DNA, was adapted to detect viral RNA with extraordinary sensitivity. Genomic sequencing of viral variants, tracking the emergence and spread of Alpha, Delta, Omicron, was made possible by decades of investment in molecular biology and bioinformatics. And the vaccines. The mRNA vaccines, developed by BioNTech and Moderna in breathtaking time, were the culmination of decades of work on mRNA biology, lipid nanoparticle delivery, and the fundamental immunology that Pasteur had pioneered a century and a half earlier. The mRNA vaccine story deserves its own moment. The idea of using synthetic messenger RNA to instruct human cells to produce a viral protein, triggering an immune response without any actual virus, had been pursued for decades. And one of the most important figures in this story is Katalin Karikó, a Hungarian-American biochemist who spent years working in relative obscurity at the University of Pennsylvania. Karikó was demoted by her university, lost her funding repeatedly, was told by colleagues that mRNA therapeutics would never work. But she persisted, because she believed, based on the biology, that modified mRNA could evade the body's innate immune defenses and function inside cells without causing dangerous inflammation. Working with Drew Weissman, an immunologist at Penn, she developed the key modification, pseudouridine substitution, that made therapeutic mRNA possible. When the pandemic arrived, their foundational work made it possible for BioNTech and Moderna to design, test, manufacture, and deploy effective vaccines in under a year, a timeline that shattered every previous record in vaccine development. Carrico and Weissman were awarded the Nobel Prize in Physiology or Medicine in 2023. It was a triumph of persistence, of basic science, and of the deep understanding of molecular biology that microbiology had built over generations. And now, as you lie there, everything we've talked about tonight is happening, quietly and invisibly, in and around you. Your immune system is patrolling, shaped by billions of years of microbial warfare. Antibodies in your blood are the descendants of molecular strategies first evolved by organisms in ancient oceans. The bacteria in your gut are dividing right now, some of them every 20 or 30 minutes, producing new generations while you rest. Bacteriophages are infecting some of those bacteria, injecting their DNA, commandeering the molecular machinery, lysing the cells, releasing new phages to begin the cycle again. A war is being waged on a battlefield too small to see. A war that's been continuous for billions of years and will outlast every empire, every civilization, every species of large animal that has ever lived or will ever live. Evolution is happening inside your body tonight. Mutations are occurring in bacterial DNA with every division. Plasmids are being exchanged. Genes are being transferred. Selection pressures are being applied. The microbial communities in your body tomorrow morning will be measurably, quantifiably different from the ones that exist right now. That's the microbial world. Ancient, invisible, unimaginably diverse, and woven into every aspect of your existence. From Leeuwenhoek's first astonished glimpse through his tiny lens in Delft, to Pasteur's swan neck flasks in Paris, to Koch's meticulous postulates in Berlin, to Fleming's moldy petri dish in London, to Avery's transforming principle in New York, to Watson and Crick's double helix in Cambridge, to Wose's redrawn tree of life in Illinois, to Doudna and Charpentier's molecular scissors in Berkeley, the story of microbiology is a story of humans slowly, painstakingly, learning that the invisible world is not marginal or secondary, but fundamental, that the smallest forms of life are, in many ways, the most important, that we are built by microbes, sustained by microbes, threatened by microbes, and armed with knowledge gained from microbes, all at once, all the time, in a relationship so ancient and so deep that it predates every other story humanity has ever told. And maybe that's a good thought to carry into sleep, that you're not alone. You never have been. You're a community, a collaboration, a conversation between your human cells and your 39 trillion microbial partners that has been going on since long before your species existed. The microbes were here first. They'll be here last. They were here when the first cell divided in some warm, shallow sea, 3.5 billion years ago. They were here when cyanobacteria filled the atmosphere with oxygen and transformed the chemistry of the entire planet. They were here when the first multicellular organisms fumbled into existence, and when the first fish dragged itself onto land, and when the dinosaurs rose and fell, and when your distant ancestors picked up the first stone tools and looked up at the stars and began to wonder. The microbes were there for all of it, and they're here now, in you and on you and around you, doing what they have always done, quietly, invisibly, ceaselessly, running the world. So let that settle in, let it drift, and let the ancient partnership that lives inside you carry on its work, as it has been doing for far longer than anyone has been awake to notice. The microbial world doesn't need your attention to function. It carries on regardless. In the dark, in the quiet, in the spaces between your cells and the folds of your intestines, and the warm, moist surfaces of your skin. But knowing it's there, knowing the story of how we learned to see it, how centuries of curious, stubborn, brilliant people slowly pulled back the curtain on the invisible kingdom, that's something worth having. And it'll still be there in the morning, under every microscope, in every drop of water, in every breath of air. Sleep well. The microbes have got this.